Hi, my name is Sylvain and I represent you PLoT-ME, which is a wrapper for classifiers to scale them to larger databases. The project lies on one side on long reads with nanopore and PacBio that has much longer reads and at a low cost. The reads now spans thousands to tens of thousands of base pair and allows for more precise identification. On the other side, you have reference genomes, and with a growth that is exponential, we have reached 100,000 of genomes for RefSeq. While the number of genomes should be a benefit for the classifiers, it's usually more a struggle for them to scale to this size of data. There has been quite a lot of work done to compress and to have better memory efficiency with Burrow-Wheeler transform, the Bloom filters, minimizers, min hash. But all of these methods still take the whole database at once to try to compress it in some ways. We propose here to have a different approach, which is the divide and conquer. Now we have our database. We need to divide it to find some features to be able to cluster it into n bins. The first part to divide could be done by the phylogeny and to cut it at some rank. It has some limitation as some genomes are much larger, and also some path of the phylogeny will have much more genomes than others. So we decided to not use the biology part, but to split genomes. We went to one step further where we divided the genomes into segments. So each genome is divided into multiple windows. Then for the features, this is a work done with Philopedia that is based on short chimeras. They have chimeras of k equal 3, 4, 5, 6, and it can classify to a taxonomy. So their work is not very precise as it cannot do it to the species or to the strain, but the advantage that it can work on untrained genomes. It also has the advantage that it's fast. And what we also saw that the spectrum, the camera spectrum changed over the genomes. And that was one of the motivations to have segments of genomes. So we can treat the repeats or the genes or the homopolymer parts of a genome differently because we have those segments. Now we have the features. The last part is for the clustering. What we wanted is something that is scalable because the size is growing fast and can do both the clustering and the classification at the same time. The mini batch is the best option that we had. So how does it work? We have all the genomes and we split them into segments. Once this is done, we compute the camera spectrum and we cluster each of the segments into clusters. We have just shown the first two, but just think that all the genomes, each of their segments can be clustered into different bins. Then once this is done, we use existing classifiers to build their indexes. So each of the index is built on a subset of the entire database. So here we have our three parameters: S for the size of the segments, K for the chimeras, and B for the number of bins. So that allows us to have much smaller indexes at once. Now that this preprocessing step is done, we can do the classification. When we have a set of DNA reads, we do the same approach. We compute the camera spectrum, and then we pre-classify those reads into the right index. Then the classification happens with a subset of the database with just one index at a time, and we have the report that we just need to emerge. So in this way, we only load a part of it with smaller memory requirements. We benchmark PLoT-ME against Kraken without the clustering. So we normalized all the results of the area under the precision recall curve with the default settings. Well, so what we have seen that K and S has a limited impact on it. Then we wanted to push the things further, so we built some communities with shorter reads. So on those communities, we have seen some trade-off, where depending on the size of the segments, so for S, we can have different accuracy. We are working on it with overlapping clusters to overcome those limitations. And the main point of PLoT-ME is to reduce the memory need. We are at 33 gigabytes with Kraken, with a whole set of microbial species. But with PLoT-ME, with 10 beans, we are down to 6.6 for the largest bean. And it goes all the way down to 2.1 with 50 beans. The beans are not equal because of k-means, but that's something we are working on. So PLoT-ME is compatible with any classifier that is capable of building its own index on a set of genomes. So we have tested with Kraken and with Centrifuge. It's a work in progress now to scale it to the whole RevSec to be able to handle any size of data. So we evaluated the accuracy, and what we saw that the identification of a species remains high. That's on the upper part of the plot. For the estimation of the abundance of the species, it gets off. That sounds something we are working on. So we now have a working prototype that is capable of reducing the memory need. What it means is that it can enhance any of the existing classifier and can scale them up to be able to look up for much more genomes and to identify more species. You can also scale them down in the sense that you can run them on a laptop or to run them on a lower hardware. You can find more on GitHub, and thank you for your attention.